Hello students, welcome to BioSmart Academy. In today's lecture, we are going to discuss about the third part of the plasma membrane. Okay, and in this lecture, we are going to start the protein part. I already covered the total lipid part of the plasma membrane. Okay, so in today's lecture, we are going to discuss only the protein part, and this protein part is very important from CSIR, NET, GATE, DBT, from any examination point of view. This protein part very important. And also I taught you in the previous two lectures of the plasma membrane the lipid part. If you don't still now if you still not see that lecture, then please go and watch that two lectures on the lipid part. That two lectures are also very important from cell biology point of view as well as from biochemistry point of view. When I taught you lipid, you will find all these things as well. So before I start this, I just want to remind you that BioSmart Academy will provide you free online lectures strictly according to the CSIR NET syllabus. And if you share BioSmart Academy and remain with BioSmart Academy, then within the 6 month of period, you will in a position to qualify any of this examination. Okay, NET, GATE, DBT or ICMR. So please share BioSmart Academy as much as possible with your friends, and please subscribe this BioSmart Academy channel and support us. Okay, so let's start today's lecture. See, today we are going to start the protein part of the plasma membrane. Okay, so basically in plasma membrane there are basically what membrane proteins are present. Okay, membrane proteins are present, and these membrane proteins are basically responsible for most of the dynamic process that are carried out by the membrane. Okay, these membrane proteins are responsible for most of these dynamic processes. Okay, now these membrane proteins are basically two types. One is peripheral membrane protein and one is integral membrane protein. Okay, one is peripheral and one is integral. Okay, please remember that peripheral membrane protein are those proteins which are basically located on the periphery. Periphery means that is not anchored to the depth. Okay, for example, if I draw a diagram, then you will understood this peripheral protein very clearly. Okay, if I draw this plasma membrane, okay, see, I draw this plasma membrane. Okay, plasma membrane, and this is plasma membrane. Okay, and this is the tail part, which is basically hydrophobic in nature. Okay, hydrophobic tail part. Okay, hydrophobic tail part. Okay, so basically I want to make you clear that peripheral protein are located on the periphery. Okay, now this protein, okay, this is the head part, and peripheral protein is located just on the head part. Okay, this is the peripheral protein which was this. Okay, this is the peripheral protein. Okay, so here peripheral protein is located, that is just periphery. Okay, it was not anchored into the lipid bilayer. Okay, so this is all about peripheral protein. Okay, and one thing remember please, that mainly most of the peripheral protein are soluble in aqueous solution. Okay, in aqueous solution, soluble in aqueous solution. Okay, soluble in aqueous solution. So please remember these 2 points. Okay, and another one important point, as this peripheral protein. Okay, so I discussed this, and another important point, as this peripheral protein, as this is located on this periphery. Okay, this located on this periphery, that's why this can be easily extracted. Okay, this can be easily released or easily extracted because it is located at the periphery, and it can be easily extracted by high or low ionic strength, high or low ionic strength. Okay, high or low ionic strength. Okay, ionic strength, so it can easily extract it. So this is all about the peripheral protein. Okay, and when we read the PPT, we see lots of more things about the peripheral protein. Okay, so let's start first peripheral protein in details, then we see the integral protein. Okay, So let's start peripheral protein. See, as I already told you that these protein are basically bound to the membrane either by electrostatic and hydrogen bond. It means here is so that the bonding is not so strong. It was basically bonded by either electrostatic or hydrogen bonding. Okay, so bonding was not so strong. Okay, second point, this protein do not interact with hydrophobic core of phospholipid. Okay, as I already tell you that basically this protein are not anchored onto the lipid bilayer, and that lipid bilayer, that is the tail portion, is basically the hydrophobic, so it does not interact. Okay, does not interact with the hydrophobic core, okay, of the phospholipid. Okay, so it does not interact, so it does not interact with the hydrophobic core, of the phospholipid. Okay, it just remain at the hydrophilic layer. Okay, so now third point, these proteins are basically bound. Okay, these are basically bound to the membrane. Okay, to the membrane indirectly with the integral membrane protein and directly by the lipid head group. Okay, I already discussed in the figure that it basically interact with the lipid head group. Okay, lipid head group, that is, if this is the head group of the lipid, then the peripheral protein is basically present here. This is a peripheral protein that is in black. Okay, it does not enter like this into the lipid bilayer, does not anchor into the lipid bilayer, just it was present in the this layer. Okay, this is the peripheral protein. Clear? Now fourth point, these proteins can be released easily by gentle extraction such as high or low ionic strength and extreme pH. Okay, this protein can be easily released. Why it is easily released? Because this protein are basically present at the periphery. Okay, present at the periphery, that why is easily extracted by using low ionic strength and extreme pH. Clear? Next point, most of the peripheral protein, as I discussed earlier, that soluble in aqueous solution. Okay, I already discussed this point, that most of the peripheral proteins are soluble in aqueous solution. Okay, I already discussed this point. Now the most important point is came here, that is the examples. Okay, please note it down. Spectrin, ankyrin. Okay, these two proteins, spectrin and ankyrin. Okay, these two proteins, spectrin and ankyrin, are common example of the peripheral proteins which are present in the membrane of RBC. So please note down the example, spectrin and ankyrin. If a question has been asked in the examination that give an example of peripheral protein. Okay, so the example of peripheral protein is spectrin and ankyrin. Okay, these are the example of peripheral protein which is present in the membrane of RBC. Okay, and what is the function of these two proteins? It basically helps in determine the shape and flexibility of the RBC. Okay, so this is all about the peripheral protein. If you still have any doubt in your mind, then please ask me the question on the comment box. Okay, I want to thanks one of my student who basically comment me on every video. Okay, and I'm very happy to see that many of the students are watched my video and they liked my video. Okay, so please watch it till end. Okay, now let's start integral membrane protein. Okay, integral membrane protein. Okay, see students, this topic is very important from CSIR point of view, so please study this topic from my videos, as I provide you in a very compact manner so that you don't have to go here and there for study. Okay, just listen to this video, note down the important points and understand the concept, and if you don't understand anything, comment on the comment box. Okay, now integral membrane protein. See, integral membrane protein. Okay, let's start integral membrane protein. So basically integral membrane protein means what? See, as we look about the peripheral protein, now integral membrane protein are basically like this. If we draw the plasma membrane bilayer, okay, if we draw the plasma membrane bilayer, okay, this is the hydrophilic part, this is the hydrophilic part. Okay, and on the hydrophilic part the peripheral membrane proteins are present. Okay, as I already told you, and this is a hydrophobic part. Okay, the peripheral membrane proteins are not anchored to this hydrophobic part, that is the into the lipid bilayer. Okay, into the lipid bilayer, peripheral membrane proteins are basically present on the hydrophilic part. Okay, okay, see, this is the basically hydrophobic part. Okay, so basically one thing, please note it down, that transmembrane proteins. Okay, transmembrane proteins are basically the integral protein are basically the transmembrane protein. Okay, all transmembrane proteins are basically the integral membrane protein. Okay, see, this is the hydrophilic part and this is the hydrophobic part, as you know. And if we draw like this, okay, if we draw like this, so this is the basically in red you can see this is the integral membrane protein. Okay, this in red color is the integral membrane protein. Okay, integral membrane protein. Okay, integral membrane protein, or you can say this is the transmembrane protein. Okay, what? Transmembrane protein. Okay, so this is the integral or transmembrane protein. Okay, now see once again the peripheral protein. This peripheral protein I draw like this. This is a peripheral protein. Okay, which was drawn in blue. Okay, which was drawn in blue. This is a peripheral protein. Okay, see yellow. This is a peripheral protein. Okay, which was drawn in blue. This is a peripheral protein which is basically loosely attached with the integral membrane protein. Okay, loosely attached with the integral membrane protein, and it's only present in this hydrophilic layer only. This, okay, this hydrophilic layer only. Okay, and this is the integral membrane protein. Okay, this is the integral membrane protein. This is the integral membrane protein. Okay, so as you can see that integral membrane protein basically cross the lipid bilayer very easily, so that's why it is known as basically transmembrane protein. Okay, and in case of peripheral membrane protein, these are the peripheral membrane protein. Peripheral membrane protein, these are basically loosely attached with the integral membrane protein, and it is mainly present on the periphery of the hydrophilic layer. Okay, this is the peripheral membrane protein. Peripheral membrane protein. Okay, so now you have a clear idea that what are integral membrane protein, what are peripheral membrane protein, why integral membrane protein are called transmembrane protein, and how integral and peripheral membrane protein basically they are present there in the bilayer, plasma membrane bilayer. Okay, so this is all about the proteins. Okay, now let's read some PPT about the integral membrane protein. Okay, now I already read out about the peripheral membrane protein. Okay, so basically integral membrane protein are protein that are held in lipid bilayer very tightly, as I already mentioned, very tightly. Okay, because they are basically anchored, as you have see on that figure. Okay, anchored. Okay, and cannot release easily by gentle extraction. Okay, like high or low ionic strength, which was in case of peripheral protein. Okay, cannot release easily by gentle extraction procedure. Okay, which was basically we have seen in the case of peripheral protein, that it can easily extracted by the use of high or low pH or high or low ionic strength. But this is not in case of integral protein. Okay, it was not going to be released easily. Okay, so that's why it is called basically integral membrane protein. That is, it is integrated with the membrane, with the plasma membrane. Okay, with the lipid bilayer. Okay, now as I already mentioned that mostly these are the transmembrane protein. Okay, these are the transmembrane protein which have one or more segment that are embedded in bilayer. Okay, these are mostly transmembrane protein which have one or more segment which are basically embedded in the phospholipid bilayer. Okay, so these are transmembrane protein. Clear? Now basically how this protein are basically embedded or basically anchored. Basically transmembrane proteins contain non-polar amino acid residue. Okay, see, the lipid bilayer is basically hydrophobic in nature, and for the encouragement you have to be non-polar amino acid, you have to contain non-polar amino acid. Okay, so basically it contain non-polar amino acid, and with its non-polar amino acid residue with hydrophobic side chain interact with the fatty acyl group of the bilayer. Okay, it means the transmembrane protein have a non-polar amino acid. Okay, and with a hydrophobic side chain residue, and with that it interact with the fatty acyl chain. Okay, if you see the figure, if you see in the figure, see this is a lipid bilayer. Okay, this is a lipid bilayer. Okay, this is a lipid bilayer. Okay, suppose this is a lipid bilayer. Okay, this is a lipid bilayer. Okay, and this is the hydrophilic layer. Okay, this is a hydrophilic layer. Okay, this is a hydrophilic layer. This is a hydrophilic layer. Okay, this is a hydrophilic layer. Okay, this is a hydrophilic layer. See, the transmembrane protein have non-polar amino acid. Okay, non-polar amino acid, non-polar amino acid. Okay, non-polar amino acid, and this non-polar amino acid have what? Hydrophobic side chain. Okay, hydrophobic side chain. Okay, hydrophobic side chain, or hydrophobic side chain. And with this hydrophobic side chain is basically attached with this fatty acyl chain. Okay, it basically attached with this fatty acyl chain, and we have fatty acyl chain, and with this fatty acyl chain it easily basically integrated, you can say, or anchored within the bilayer. Okay, so in this way integral membrane protein basically anchored. Clear? If you still have any doubt, then please comment. I will make you clear on the comment box. Okay, next, see, and one more important point is that all the transmembrane proteins are amphipathic. All the transmembrane proteins are basically amphipathic in nature. See, amphipathic means it contains both hydrophilic as well as hydrophobic layer. Okay, so that's why these are amphipathic. As you already see in the figure, it crosses hydrophilic to hydrophobic layer. Okay, next. Now, these transmembrane proteins is of two types. One is single pass, which is called monotypic, and one is multipass, which is called polytypic. Okay, transmembrane protein are classified into two types. Classification is two types. One is single pass, one is multipass. Single pass is called monotypic, and multipass is called polytypic. Okay, now the example of a single pass or monotypic transmembrane protein, the example of a single pass or monotypic transmembrane protein is glycophorin. Best example of a single pass or monotypic transmembrane protein is glycophorin. Very important from examination point of view. Note it down. Glycophorin. And what is glycophorin? Basically, it is an protein which is present on the RBC membrane. Okay, and it is a single pass transmembrane protein or monotypic transmembrane protein. Okay, basically glycophorin is a small glycophorin is a small glycoprotein which contain 131 amino acid residue. Okay, and first membrane protein which have the complete amino acid sequence. Okay, but still the exact function of glycophorin is unknown. Okay, so if a question is been asked in the examination that give an example of an single pass transmembrane protein, so the answer will be glycophorin. Next, now give an example of an multipass or polytopic transmembrane protein. So multipass or polytopic transmembrane protein is band 3 or chloride bicarbonate exchanger protein. Okay, it's an example of multipass transmembrane protein. I put it on red color because it is very important from examination point of view. Okay, so band 3 or chloride bicarbonate exchanger, band 3 or chloride bicarbonate exchanger is an example of multipass transmembrane protein. Please note it down. Clear? Now you know very well that chloride and bicarbonate exchanger is basically when one chloride molecule is inserted into the bilayer, bicarbonate is basically out, and when bicarbonate is inserted and chloride is out. Okay, so in this way this mediated electroneutral exchange of chloride and bicarbonate. Clear? See, this point is very important. Okay, see, one of the main characteristics of transmembrane protein that it have a alpha helix. See, for future perspective of the cell biology, when I going to teach you cell biology in very much depth, okay, in very much depth, then all these things are came. Okay, so you can understand, so you can clear all these things now, because these are the basics of cell biology now. Okay, see, transmembrane alpha helix, this is the basic thing. So you have to know that transmembrane protein contain alpha helix, and each alpha helix have 21 to 26 hydrophobic amino acid residue. Okay, and this hydrophobic amino acid residue help to facilitate the spinning of bimembrane layer. Okay, means transmembrane protein are basically have alpha helix, and this alpha helix contain 21 to 26 hydrophobic amino acid residue, and this hydrophobic amino acid residue help in spinning the membrane bilayer. Okay, if you clear this with the help of this diagram, see, this is transmembrane protein. Okay, this is transmembrane protein, this is transmembrane protein. Okay, this is transmembrane protein. Okay, this is transmembrane protein, and transmembrane protein contains alpha helix. Okay, what? Alpha helix. Alpha helix. Transmembrane protein contain alpha helix, and alpha helix contain 21 to 26 hydrophobic residue. Okay, hydrophobic residue, and this 21 to 26 hydrophobic residue helps in spinning membrane bilayer, helps in anchoring membrane bilayer. So now you have got a clear idea about this transmembrane protein, Okay, next. So this is all about the transmembrane protein. Okay, now in few membrane protein you have been seen that transmembrane portion comprised of beta barrel. Okay, what is beta barrel? Basically beta barrel is made up of anti-parallel beta sheet. Okay, this beta barrel is made up of anti-parallel beta sheet. Okay, I will teach anti-parallel beta sheet during biochemistry section. Okay, during the biochemistry section. Okay, that what are anti-parallel beta sheet. Okay, so just remember this thing, that this beta barrel is made up of anti-parallel beta sheet. Okay, anti-parallel beta sheet, this present in some cases in few membrane protein. Okay, this transmembrane portion comprise beta barrel, which is made up of anti-parallel beta sheet, and the most common example of beta barrel which is made up of anti-parallel beta sheet, please note it down: porine. Okay, what is the example? Porine. What is porine? Porine means, as you know from the name, that is porine, that is a pore forming protein. Okay, which allow hydrophilic solute to diffuse. Okay, it is an pore forming protein which basically allow the hydrophilic solute to diffuse. So this is about the porine, and porine is an example of beta barrel protein that is made up of anti-parallel beta sheet. Okay, made up of anti-parallel beta sheet, and these beta barrel are basically, as it helps in diffusing hydrophilic solutes, so that's why it made up of hydrogen bonding between the beta strands. As we know that in anti-parallel beta sheet, these strands are basically that present, and bonding is H bond. Okay, bonding is H bond. Okay, bonding is H bond, that is hydrogen bonding. Okay, clear? So the interior of the barrel is hydrophilic. That's why it's allow hydrophilic solute to diffuse. Okay, so if we look at this, that this is a basically some transmembrane protein contain beta barrel. Okay, contain beta barrel. And this beta barrel is made up of anti-parallel beta sheet. Okay, anti-parallel beta sheet, and one example of this anti-parallel beta sheet is porine. Okay, this porine, P-O-R-I-N, porine, and this is an basically nothing but a pore forming protein. Okay, this is an pore forming protein. Okay, and this pore forming protein is helps in diffuse of the hydrophilic solute. Okay, so students, that's all for today's lecture. If you like this lecture, then please share as much as possible and subscribe Biosmart Academy. Okay, I try to explain in a diagrammatic way every PPT I try to explain in a diagrammatic way. Okay, if you got an explanation like this in a diagrammatic way, then this will make your concept more and more clear. Okay, so if you like my video, if you have any doubt, then please comment and please share as much as possible. Thank you.